Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is October 10th, 2024. We're talking with Ranmal Samarasinghe. Ranmal,
Hi.
did I get that close?
You did. Thank you.
Okay, good. Ranmal is assistant professor in the Department of Neurology at the David Geffen School of Medicine at UCLA. He's a practicing neurologist with a clinical interest in epilepsy. He's also an experimental scientist working on changes in cortical circuitry that are associated with epilepsy, and maybe other things. That's what I'm focusing on my reading. So his research focuses on some well-known genetic mutations that produce developmental epilepsy in humans, and he uses cortical organoid models, which can be grown from stem cells derived from the human patients so that they are good models of that particular human disease. He uses a variety of methods to study these, to study their development, their structure, the cell types that make them up, synaptic connections, network function of these little pieces of human cortex grown in vitro. So welcome, Sara.
Thank you.
Also with us is Sara Mirsadegi.
Yes.
And
Hi.
Sara. Sara is a PhD student in our neuroscience program at UTSA and is one of our local cortical organoid experts. So thanks for joining us.
Glad to be here.
And I'm your host, Charlie Wilson. So, our brain organoids are relatively new development that have progressed rapidly, and the capabilities of brain organoids seem to be expanding all the time. And our listeners are probably familiar with the idea, but could you fill us in a little bit on the basic technology and especially the kind that you like to work with? And then we'd like to hear something about the value in studying human genetic disease and development, and also how we can use them just to understand the development and function of the cortex.
Sure. So what is an organoid? So organoid is derived from stem cells, and that can be induced pluripotent stem cells, so from patients, or embryonic stem cells. And they're 3D brain-like structures. And so the basic idea is you take the stem cell and you differentiate them in a very particular way using particular morphogens, and you direct them towards a certain brain-like region. And the advantage is that compared to 2D culture, and as an in vitro system, you're able to see a more sophisticated, layered cytoarchitecture. So just like cortex in brain has layers from progenitors to the upper layers, including neurons, you can see this developing in the organoid itself. And this lends itself to some, in our hands, some electrophysiological activity that's more sophisticated than we'd expected. We were able to see electrophysiological activity like we can see sometimes in recordings from ex vivo mouse brain, for example, or even in vivo human recordings. Now, our lab in particular makes these things called assembloids, which are a slight variation on organoid, but the assembloid, the idea is we fate specify early in development, so which is to say we'll make an organoid that is representative of a certain kind of brain region, let's say the cortex, and then an organoid that's representative of a different kind of brain region, and we put them together. That is the assembloid part. So we fuse them. And the main rationale for doing this really is that we want to have a structure, an in vitro structure that has an adequate diversity of cell types, excitatory cells, inhibitory cells, and glia, so that we can get some robust electrophysiological or electrodynamic activities. So we actually studied cortex, and we do a lot of work in hippocampus as well, but the same idea, we put these two things together. We put the hippocampus or the cortex, it's mostly excitatory cells, with a part that mostly generates inner neurons. We put them together to make the assembloid. We see migration of those inner neurons to populate the mostly excitatory part and generate this kind of more diverse structure. Then you asked about what we can learn from this as a developmental model. Well, I think the obvious advantage of the model that we can leverage is we can get these iPSCs from patients who have particular diseases. And so in my case, I largely am interested in epilepsy and autism, and I see patients who have severe epilepsies, including single gene carriers of single gene variants that result in epilepsy. So we can take blood, for example, from these patients, and we can make an induced pluripotent stem cell. And from that, we can make an organoid. So in that way, we have sort of a replica of the patient's brain in a dish, in a way of saying it, right? And then we can look at activity. And as a developmental model, it's also good because it's human. There are differences between, for example, human brain and the brains of non-gyrencephalic mammals that are important, that may be important in development. And we can, and the organoid captures some of these differences. For example, basal radial glial cells are not so common in lissencephalic mammals, but very, very highly expressed in human brain and are important for the massive expansion of the human cortex. And we see a lot of basal radial glial cells in organoids, in human brain organoids. We see an outer subventricular zone, which we don't see in lissencephalic animals. And so on and so forth. And so, in this way, I think there's aspects of development that we can model. And the organoid seems to generally track with fetal development. And so we can look at, if we're looking at a 3 to 4 month old organoid, there are many parts that may be similar to 3 or 4 month developing brain, in a fetal brain.
And of course, I guess it's obvious that for studying a human disorder, you're studying the actual human disorder.
Yeah, exactly. So, yeah, and that was just sort of what I was trying to get at, and then we can get these cells from the patient. But yeah, that goes without saying that we can get the specific variant or whatever that is in the patient, and in situations where perhaps you have to artificially express this in a mouse because mice don't express these genes, or in the case of the organoid, we don't have to do that. So what's your sort of vision on using this to understand potential treatments for humans, or something about the development of the disease? I guess the idea is that this is a sort of better approach to develop future treatments than, say, trying to make a mouse that has the same disorder, or at least complementary. Yeah, and that's a key. I don't think it's better. I think it is complementary, and we have to use both approaches. There's certainly, it is a reductionist system. There are limitations, but there are advantages. Some of the advantages we've described, but it is an in vitro system that's highly manipulable, and there are certain things that we can do relatively easily. For example, if we want to understand the type of cell that's contributing to the disease pathology, when we're making these assembloids, a kind of a relatively simple experiment in an organoid that's much more complicated in a mouse, is we can take one part that has the mutation, and then we can take another part that doesn't. So, for example, we can have a cortical organoid that's mostly excitatory cell that carries whatever mutation we're interested in, and then we can have a ganglionic eminence organoid that's mostly in a neurons that doesn't. And in this way, we can understand what cell type out of these two broad categories is primarily responsible for the pathophysiology. And it's harder to do in a mouse, and we can do that relatively easily in an organoid. In terms of thinking about, for example, personalized medicine, there's really no other option in some ways, right? This is the most sophisticated in vitro system we have. And in the case of some of these rare genetic disorders like the ones I work on, it almost makes economic sense to, at some point, if we can get to that level where we can generate organoids from a particular patient and, for example, test drugs. And I'll give you an example. So in epilepsy, how do we decide what anti-epilepsy drug or what anti-seizure medication, which we call it now, what anti-seizure medication to give? Well, we have some rubrics. We have some ideas. If it's a sodium channel mutation, you want to give certain kinds of drugs. You want to avoid certain kinds of drugs if it's a gain of function. But there's as much art as there is science to this. And we'll give a medication. Hopefully it'll work. Sometimes it doesn't. Sometimes the side effects are bad, etc. And sometimes the medication works and then it stops working. And then what do we give next? And this is while this is happening, the patient is having seizures. The patient is still sick. It would be really nice if we can predict what medication is going to work. And this is knowing that there's probably out of our large armamentarium of medications, there's Now, probably there's definitely better and worse, and probably some that are going to be really good. But how do we get there? Well, we guess right now. We don't guess. It's an educated guess. And sometimes we also have a lot of trouble because a patient starts getting better, and we pull off a medication thinking that this is not really helping, plus their side effects, and suddenly the patient gets a lot worse. Then we put the medication back on, now it doesn't work. And that's really troubling and really challenging to deal with. Now, these kinds of situations, we can imagine kind of a scenario where we generate organoids from a patient who has whatever disorder that's really rare, that's very expensive to treat, that if we don't treat well, it's going to be very expensive because the patient's going to go to the ICU and get sick and have lots of seizures, and et cetera. It wouldn't be really not feasible in that situation to make an organoid from those patients and try the drugs. And if we have some reliable biomarkers, understand what's working and what's not, and say, okay, you know what? For you, I'm going to start with Keppra because I know that's going to work well. Then I'm going to add clobazam because I think that's going to add to that. I'm never, ever going to give you Depakote because whatever reason, et cetera. And I know that if I pull off this one, then I should add this one, et cetera, if there's a side effect problem.
So all that iteration and titrating doses and everything could be done in a dish.
Yeah, potentially.
Or at least some of it.
Some of it, maybe.
Just some of it.
Yeah, and even that would be an advantage, for patients who are really suffering and where we're having a lot of trouble taking medications. Right?
And then there's a potential for a lot of basic science advance. So one of the things you mentioned is just the ability to know whether the mutation is mainly affecting one cell type or another. And it's very common for people to say they think that what's wrong in seizures has something to do with interneurons. So which is it? I mean, is it the interneuron? You've done this experiment, at least for the couple of mutations, which...
We have, and the answer is it's not always that, right? And that's interesting. So in early studies that I did as a postdoc in Rett syndrome, where it's not real, I wouldn't call that a bona fide seizure disorder, but these patients that we got, the iPSCs, had seizures, or at least epileptiform abnormalities and autism. There, it seemed like it wasn't a neuron problem. Clearly, from the experiments that we did, the kind of the precise experiments I described. More recently, in my own lab, we've been studying a sodium channel mutation, SCN8A, that is actually a bona fide epileptic encephalopathy. It's called Developmental and Epileptic Encephalopathy 13, severe seizures in most cases. There, in the cortex, actually, it seems to be more of a glutamatergic problem, that you get more glutamatergic cells. I mean, that they seem to differentiate and you have more of them. You don't see really a loss of interneurons, but maybe that's resulting because there's more excitatory cells, there's an EI imbalance there. And otherwise, it might just be that you have this gain of function channel and it's just more sensitive and fires more, and so it's hyperexcitable. So we don't think it's an interneuron problem. In the same variant, we also looked at hippocampus, and there we also saw an excitatory cell kind of a change in the kinds of excitatory cells that were expressed, but also a reduction in specific interneurons, right? So it's complicated. It may be regionally specific. So cortex may be different than hippocampus for the same gene. And certainly for different genes, it may be very different. And we can examine these things using these models.
And those differences are probably developmental. And so having a model that allows you to study the development in a really direct way. If you have to do a longitudinal study of development of the organoid, you could find yourself killing a lot of organoids, but that's quite a bit easier than killing a whole lot of mice.
Exactly.
To reconstruct it.
Right. Yeah, and I think in that sense it can be efficient, and we try to generate 100 organoids at a time if they survive. But that's when we're plating, that's what we're doing. And clearly, by the way, I think one of the things we're learning more and more, just thinking about this from a therapeutic standpoint, not just from my lab by any means, but just looking at the field, it seems like even in the case of these channelopathies, like SCN8A is a channel problem, there clearly seems to be something that's happening in fate specification in the differentiation of the cells. And we're picking these up in organoid models for certain, and in thinking about therapies, we probably have to address this somehow if we really want to help these, if we really want to achieve something approaching a cure. And once again, even that, or these are experiments that we could potentially trial first in an organoid model.
Yeah, and potentially find a developmental lever that could be applied at one point in time and change the course of development.
True, correct.
That would be.
You're
not promising that, but that's like a good thing. So one of the things that impressed me is the possibility of addressing like some long-standing questions about cortex in general. So, for example, you see these cortical oscillations over pretty much the entire frequency range that we're used to seeing in humans, and you have human EEGs you can compare to what your organoids do, and they look really similar. Mm-hmm. But for a long time, it was thought that the thalamus was really critical for some of these oscillations, and you don't have a thalamus there. So it seems to me that there's, of course, that doesn't mean the thalamus isn't part of the oscillation, but it seems to me the possibility for maybe asking what is the minimum circuit required to create some of these oscillations, and also even though the corticoid gets to layers and cell types, it isn't really as layered as a regular cortex, and some of the details are probably a little different.
Yeah.
Right? But maybe those things don't prevent the generation of oscillations, so you could end up with a sort of purified system for studying those.
Yeah, and I think, an oscillation is just that. It's an oscillation. And I even for the oscillation, I wouldn't say that the oscillation that we're seeing in an organoid is exactly even if it's the same frequency, it's the same mechanism of an oscillation in an in vivo brain. In fact, it almost certainly is not. We don't have myelination, we don't have connection. We can make a thalamic organoid and connect it, but it's not going to be connected in the same way or the same maturity as an adult or even a newborn baby's brain. But that said, I think the one way we think about the oscillation or looking at these electrophysiologic activities, is complementary to the other things that we can do. We can do transcriptomics easily, we can do immunohistochemistry easily, and we can do different modalities of looking at electrophysiology like single unit activities or calcium imaging, etc. But what it provides for us, I think, when we can see that is as sort of an indication that it's a healthy organoid in our hands, and that those little microcircuits at least have reached a certain levels of maturity, from which then we can see differences from mutes. Because it seems like what happens in a lot of these developmental disorders, or perhaps they happen also in degenerative disorders, although we haven't looked at this, is that the rate of maturity is different. In some of these developmental disorders, it looks like it's maturing more quickly, but also kind of incorrectly, like the right stuff isn't there. And the oscillation is the immediate kind of biomarker to indicate that there's something wrong. And then we can dig into the anatomy. And when we're trying to really fashion a story, we want to have all these components, right? And so we're not going to say this oscillation is the human oscillation. We're just saying it's oscillating at these frequencies, which means that there have to be some of these cells that are important to generate these, at least in this local area. That's all. And then we can say, okay, if there's a difference, now we can start looking at other ways of examining the structure to understand why. And so, there's a loss in this particular kind of interneuron, or there are these, the synaptic, if you count the synaptic puncta, that there seem to be more excitatory puncta than inhibitory puncta, or whatever. And when we look at the transcriptomics, hopefully that's complementary to that and shows the same thing. If we took all these together, then we can I think we can be more confident in saying that, this seems to be the pathophysiology of this particular change that we're seeing.
So one of your outcomes was just a loss of oscillations over a wide range of frequencies in the mutant organoids. Is that did I see that right?
Yeah. So you're talking. Yeah. So in one of in cortex in MECP2, we saw a loss of lower frequency oscillations, but we saw actually a gain of very high frequency oscillations, that we think could be like the pathological high frequency activity or high frequency oscillations that are associated with like seizures and hyperexcitability. I think that also kind of was consistent with the rest of the story, which is to say, like in the case that you're talking about, we lost low frequency, we gained some high frequency. We saw a lot of like individual discharges that looked like spikes. And we specifically lost a lot of gamma activity. And we saw this inverse relationship between the frequency of spikes and the power of gamma. I say all these things because these are some of the same things that we see in human patients with these disorders. Like we see spikes, we see reduced gamma. Gamma is associated with sometimes with certain cognitive processes too. It seems like the machinery that's involved in gamma gets kind of co-opted to generate these spikes, and so we see a loss of gamma in the human patient. Kind of saw the same thing here. So yeah, I mean, in that particular case, there was a lot of stuff going on, but I think it just overall it looked like the circuits were very disordered. But we don't always see that. We see other situations where the oscillations seem to be there, but more subtle changes occur in the interactions of oscillations.
Of course, the things you're calling spikes are like giant, momentary, synchronized events. And then it looked like lots of cells were firing in a fairly narrow time window, and then they would go silent sort of for a long time. So that's very organized. It's just like organized in a terrible way for the cortex to work right.
Yeah.
So, as you know, for somebody studying circuits, it seemed like a big opportunity to go in there and, you know, ask what individual neurons are doing. And one thing that I don't see a lot of in the organoids is that people going in and doing intracellular recording on identified neuron types or anything like that, which we do it a lot in slices. Why is that? Is that hard?
No, I mean, it is hard. It is hard, but the reason isn't because the reason I don't think it's because it's necessarily any harder than what you're doing. I think it's largely historical. If you think about organoids, who are the people who are really doing organoid work? Who are the big names who are like publishing in the top journals? They're mostly stem cell biologists, developmental biologists. The questions they're asking and that they're interested in are not these questions, like overwhelmingly. There are very, very, very few of us. Even I'm not trained as a physiologist, actually. I came into this, I'm a clinical neurophysiologist, and so I started doing this as a postdoc, really, and I'm only doing local field potential recordings. But I think that has a lot to do with it. The folks who want to make the organoids and who make them well and have the resources to do a lot of experiments are doing CRISPR-I experiments, you know, on organoids. They're looking at development. They're doing these giant sequencing experiments with like ATAC-seq and single cell and blah, blah, blah all together, multi-omics. And these are the big labs. And there are a few of us who are more interested in physiology who are working on organoids. That said, just recently, and it's been hard for me because I am also not someone who's patched on my own, but in the last like month or two, we've been working really hard to set up a rig, using a collaborator's rig that he's not using, and start doing some of these basic physiology experiments. I think they're incredibly important when we need to do them, and I've asked this question not just by you, but by others. But there's a lot of questions to ask and answer, and we haven't gone to it. And I think the people who probably have the resources to do it are not really looking to do that.
So some of the things I was thinking about are modulatory inputs. You know, we think serotonin and norepinephrine are terribly critical in the cortex and important for determining the time courses of the processes that give rise to oscillations at particular frequencies. So what struck me most was that the spectrum of the field potentials looked so familiar. There were like delta oscillations, and they were clearly looked right, and theta and alpha, and so on. And so each of the normal thinking about that is that these are set by time constants of, the duration of synaptic action or the time required for a cell to integrate its inputs to fire, and that each one of those has these different time-related processes. And we know something about, the ion channels that are responsible for them. But it's really hard to go in and it would be really hard for us to go in and modify one of those ion channels so that its time constant was changed. But maybe not so hard for people who stem cell biologists, developmental molecular geneticists. So the combination of those things could be really spectacularly cool for physiology of the cortex.
Yeah. What do you mean by, so how would you mean like genetically modify the ion channel specifically or something?
Yeah.
Okay.
In a particular cell. Every one of these models, I don't know, this is just me, but every one of these models you see for how an oscillation is set up, and there are a bunch of them for all the different frequencies of oscillation. Computational neuroscientists have made wonderful models that reproduce these things and tested them to some degree. Like you can put on some drug that blocks an ion channel or something and make an oscillation go away or make it bigger. It doesn't really get to the question of what neuron is it that has the ion channel that you are interested in. And so dissecting that stuff has been kind of impossible, but the hypotheses are numerous and well-formed and turned into computational models that make predictions like the ones you have used some of that. Mm-hmm. And so the people who are interested in that, who study neurons in slices or in the whole brain, would die for some of the tools that are available for organoids. I don't know. That's just my pitch.
Yeah.
So I'm interested also in the sort of laminar structure. So your interneurons are, I guess I should say GABA neurons, because maybe they're not all strictly interneurons. But GABA neurons are migrating in from an organoid that's sort of stuck on the side. And presumably the only ones that are really helping are the ones that did make it in. But possibly not. Possibly the excitatory neurons have grown axons out into the inhibitory cells. Culture in the inhibitory cells have grown axons in. And so this raises another set of questions that we've always thought that the actual geometric structure of the cortex was important because it's so interesting and layered, and there's a parabellum cell in layer 5 and there's a parabellum cell in layer 3. I wonder if they do different things. Kind of normal question for us to ask. So what do we know about the anatomical arrangement between excitatory and inhibitory cells in the culture?
Not much, I think. Right, and that's it. And I think that is, you're getting at the limits of what our culture system can do right now. So, like, for that kind of specific question, like the PV in one layer versus the PV in the other layer, I think with an organoid, it's just the PV and an excitatory cell and some stuff around it, as opposed to in a specific layer. Yeah. The reason is actually, even though we see this layered organization during development, as we move towards the level of maturity that we need, and maturity I mean age, and I think the cells then mature enough to generate some of the oscillations that we've shown, that layered structure tends to kind of break apart. Essentially, I think what's left is an outer layer that's mostly mature cells, like deep and upper layer excitatory cells and interneurons, and then some progenitors and a lot of cells that are actually start dying off because it's the inner core of the organoid and it's not getting oxygenated enough and it becomes necrotic. So those kinds of questions, I think, we're not really equipped to answer yet.
I think in a way it is an answer. If the electrical behavior you are expecting from the cortex is there and the layers aren't, then the layers aren't responsible for that fundamental thing. I think about it because bird's cortex doesn't have layers.
Yeah.
It has all the right cell types, but they're not in layers.
Yeah.
And yet the bird's cortex works pretty well. So I guess you could try to dig in and ask, what does the bird cortex lose by not having layers? Would be a cool question, but hasn't really been addressed. But here, I think the question of what does the organoid function lose by not having layers may actually emerge as you go along.
Yeah.
We're not seeing anything that it loses so far.
Yeah. We're
Maybe
not.
later.
Yeah, that's interesting.
Yeah. And it may have to do not with the generation of these emergent sort of properties, these emergent oscillations, but maybe about where these emergent oscillations originate and feed into. Like you can imagine maybe layer 4 goes to a certain part of thalamus or whatever, layer 5 somewhere else. But that doesn't exist in an organoid that's just sitting in a dish that's not connected to anything.
I know Sara is interested in this.
Yeah.
What do you think that the organoid can actually be a computational device and it could be trained to solve problems and do things?
I'm hoping for that. So yeah, I was thinking like specifically I'm interested in information processing, and these assembloids that we generate in a dish, they already contain the basics component for information processing, which is excitation and inhibition. But like in epilepsy situation, we have hypersynchronization. So I'm thinking, and also people with epilepsy, it's not all about seizure. It might be intellectual disability or cognitive, other cognitive dysfunction too. So with other technologies that we recently have, like multi-electrode array or optogenetics, we can also feed some input to this network and see what is the response of the network to this electrical stimulation or optogenetical stimulation, and what is the difference between the information processing within that epileptic hypersynchronous circuit versus like the healthy circuit. So I think that can also the assemblies can also provide us with a like simple platform to study these kind of questions.
So one of the things you need for what we normally think of as the computational function of the cortex is synaptic plasticity.
Yeah.
So do we know anything about synaptic plasticity in the organoids? Have there been studies of?
Yes, there is actually a bioarchive study that was released in bioarchive, I think, weeks ago, that they used multi-electrode array, feeding the organoid with this electrical stimulation, and then the same organoid after the electrical stimulation, they looked for genes that are involved in plasticity. So they check those genes and they address if those genes are upregulated after that electrical stimulation.
Cool. It's not my usual definition of synaptic plasticity.
Yeah, that's true. Yeah, I agree with that.
It is a start.
Yes.
Okay. Well, thank you very much. It's been fun. Ranmal,
Thank you
for joining us.
Pleasure.
And Sara,
Thank you for having us.
this has been Neuroscientists Talk Shop.